relatively rare, and a number of them are pretty good relative to you know, what rates have been in the Corn Belt for the last 70, 80 years. They've likely increased from this level in the last few years, but there aren't recent estimates. So CIMMYT's estimated its rate of gain recently, and under favorable conditions, it's pretty good. In most of the programs for which estimates exist or have been recently generated, under more difficult condition, rates of gain, as you would expect, are pretty low. So if we are going to contribute to poverty alleviation in the most difficult environments, we have to think very carefully about how to increase this effectiveness, and it's been exciting to listen to the developments that we've been exposed to at this meeting in that area. We think that genetic gains are delivered in farmers' fields of producible, marketable varieties are the key metric that we need to be thinking about. And we're thinking pretty hard about how to help research managers understand the key drivers of genetic gain, and that was run through very nicely in some of the presentations, particularly Jesse's just before lunch. We're helping develop scorecards and metrics by which we can model best practice for cultivar development and for the programs we invest in. We're seeking ways to really help the CGIAR coordinated platforms, CG and its national partners, sustainably increase the rate of gain that they deliver. And we're also, it's not enough to generate this in our nurseries and our trials. We have to really plan for more aggressive delivery. We see a number of key challenges, and the application of genomics in cultivar development is among them. But really, I would put first improving research management to ensure that crop improvement teams in public sector programs serving smallholders in Africa and South Asia, our target environments, are really focused on and rewarded for and held accountable for delivering high rates of genetic gain. Increasing the signal-to-noise ratio in phenotyping, by that we mean heritability or repeatability, without increasing replication, is going to require investment in mechanization, increasing size of programs, increasing accuracy. We do need to increase program size, throughput, population size through mechanization and automation. And this is happening very slowly in most of our programs. A focus of what I'm thinking a lot about at the moment, and we're discussing today and in a workshop tomorrow, is how to make genotyping costs trivial relative to phenotyping costs, facilitating the application of genomic information. We've heard a lot about how genotyping is a solved problem. It's solved in theory, it's solved by a few very innovative people, but putting the logistics together, the plans, the pipelines, this is not trivial to work it out in cost-effective ways in our contexts. And of course, the objective of this is to reduce breeding and cultivar replacement cycle time as a primary driver of increased rates of gain. And the need to speed up cycles, we give it lip service, but I know in my own program, it took me a long time to be very analytical about that, and it's an area where we need a lot of improvement in many of the programs in our institutions. I don't know what I had for 0.6, but obviously I removed it. Information management is another area where very substantial improvements must be made very quickly, and the tools right now are being put in place to make these improvements. any amount of wonderful genetic analysis leading to the tagging of useful genes will not be helpful if breeding programs are operating off unlinked spreadsheets and not using integrated breeding informatics systems. If we address these areas aggressively, there are really big gains to be made. I'm inspired by what's happened in the Embrapa, the Brazilian upland rice breeding program, which has tracked its progress. This is derived from a paper by Flavio Bressegallo in Crop Science in 2011, I think. From 1984 to 1992, they documented gains of about 6 kilos per hectare per year for upland rice, or 0.2%. By increasing the program size, more than doubled those gains in the next decade. And more recently, tripled them again by reducing cycle times, increasing throughput, increasing accuracy. So there's a lot of scope for us to be analytical and aggressive about improving these pipelines, but we have to measure what we're trying to improve. So let's think specifically about genomics and how it contributes to this increased performance of the system. Genetic analysis, which is mostly what we've been talking about over the last couple of days, has delivered a number of important mass products based on genes for resistance to biotic and abiotic stresses, or for tolerance, large effect QTLs. I'm familiar with a number of examples in rice from Erie, where I used to work, for drought tolerance, anaerobic germination, submergence tolerance. And these products are moving into selection now. So I would argue that genetic gains from genomics at this point are not really discovery limited. We have a lot of products that need to be moved into these programs. They're application limited. They're production system limited. And by production system, I mean production in the breeding context, the breeding pipeline context. So there have been massive investments in crop genomics over the last 20 years, but not a lot of application in public sector cultivar development. Huge amounts of resequence and GBS data have accumulated in our programs. Hundreds of thousands of lines have been GBS'd at the CG centers, and thousands have been resequenced. But queryable databases have not been developed, and breeders are not using this information in genomic prediction applications that we heard about in the session earlier. Breeding programs in general have yet to adopt integrated database systems for managing population development, phenotyping, selection, decision support. And these have been available commercially for a while, but good publicly available open source systems are only coming on stream now. And we'll hear about the integrated breeding platforms product, the breeding management system, I think, a little later. But it's important to note that genotyping costs for public sector breeders are about 10 times higher than they are for the multinational seed companies. We think that, me and my colleagues at the Gates Foundation think that genotyping costs for Monsanto Syngenta Pioneer for a genome profile that's good enough to use on initial selection candidates are likely less than $2 a DNA sample, maybe a lot less than $2 per DNA sample. And we can't get that type of profile at the moment in the public sector unless we are Jesse Poland for less than $20. And now that's going to change fast, but at the moment it's logistically difficult. Breeders, I think, really need a low-cost genome profile if genomic data is going to be widely used and used in routine cultivar development. So when we say that genotyping is a solved problem, the logistics are the problem that remain to be solved, and they are very sticky problems. We're trying to, at the Gates Foundation, support the infrastructure for moving the results of the wonderful work that you and colleagues are doing into routine use in breeding programs through projects like the integrated breeding platform, which is going to be is now a very broadly applicable breeding informatics system for managing all aspects of population development, phenotyping, low-density marker application, selection decision support. In order to try to organize and make available the, and there are other custom, more customized applications for rice, the B4R system that IRI has developed for its own use. Cassava base, a system that focuses on, or is designed for applications in vegetatively propagated crops. In order to help organize and make accessible the massive volume of high density and resequence information, we're supporting a project called the Genomics Open Source Breeding Informatics Initiative. It used to be called, referred to as the Genomics back office, which will help individual programs organize their high-density data into queryable databases that can then be directly used by breeders in forward breeding. So it'll implement systems, databases, analytical pipelines that will bring these data into routine use. Another project that is getting started is the integrated genotyping service and support facility, which is being put in place at Becca, Biosciences East and Central Africa in Nairobi, under the leadership of Apollinaire Jeking here in collaboration with DART. And this will provide both a low-cost GBS service and a lot of support to breeding programs on actually applying the data in applications like quality control, parental diversity analysis, genomic prediction. And of course, Jean-Marcel Ribot is here and can update you on the integrated breeding platform. He's the lead of that project. The GOBII project is being led by a group at Cornell and the Boyce Thompson Institute. Susan McCouch is the PI with Ed Buckler and Jean-Luc Yannick. I'll say a few words about the genomic back office and how we hope it will help us bring genomics data into routine use. These are slides courtesy of Susan McCouch. Its aims are really to put in place the plumbing, infrastructure, and scaffolding, allowing breeders to access or to exploit high-density marker information. The project's starting with rice in a collaboration with IRI, with wheat and maize programs at CIMMYT, and with sorghum and chickpea at ICRISAT. And these crops were chosen because they have strong breeding programs, and they have very good genomic resources in terms of functional SNP pipelines and strong HapMaps with sufficient diversity information to start the work. But the plan is to add additional crops as resources are found. And the idea is really to develop a sort of a data integration ladder across, densities to allow the whole HapMap to be imputed on selection candidates using, a very small number of markers. And this is really the approach that the large seed companies are taking right now. There may be other approaches. This was the initial idea. So you may have a reference genome that costs $100,000 to generate. And once you've got that reference genome, in these crops, there are a lot of key ancestors that are resequenced in any program. It's not a really big outlay to resequence, say, 100 key ancestral lines or genotypes from the program, represented by those entities A to E there. And you would use those, trace those haplotypes down through the breeding program to parents of the current generation. They would be profiled using GBS with different flavors of that, have 20 to 30,000 to 2 million calls, an average price, say, of $30. But you can link it back to the whole HapMap and the parents. And once you've got that imputed on the parents, the selection candidates only need to be profiled for 100 to 200 markers. So but putting that scaffold together is a very large undertaking, which getting the different platforms onto one scaffold is what GOBII is going to try to do. So it'll create large databases that are easily queried, easily stored and accessed. It'll link those to pedigree databases to help with in the breeding informatics systems, like the breeding management system B4R, cassava base, so that the genotypic data then are linked to the breeders' pedigrees and the breeders' populations. Develop breeding schemes and data pipelines for selection applications, genomic selection applications, and help provide the instruction and support to get breeders ready to use these. There'll be two interacting teams similar to how these systems have been put in place, I think, in a number of the companies. One will be sort of a breeding science translation team. These will be people who are very knowledgeable of breeding methods in the crops, based at the CG centers, who identify breeders' needs, transmit ideas to developers, prioritize tool development, and a very strong software development group that will actually code the tools. The back-end functions would be a data integration tool for across these densities, imputation and phasing tools, estimation of tools for allowing breeders to track IBD blocks, haplotype blocks, and visualize them in their breeding materials. Breeding support functions would include genomic prediction tools, haplotype tracking tools, decision support, and the ability to link in both large and minor effect QTLs. I'll just skip quickly through this. The process, the visualization that Susan provides here is that GOBII will provide the back end, and the high density prediction functions that will be linked through APIs to the breeding informatics systems that breeders are adopting and using, and that these will essentially serve as the user interfaces. The second area where we really need to make progress is to reduce genotyping cost per sample. This is a very serious problem still. Genomic selection approaches depend often on high selection intensities, increasing selection intensities. They require us to genotype a lot more lines than we phenotype. And costs per sample are driven drive the applications, not cost per data point. At the moment, my understanding is that for most programs, $20 is now a pretty low cost for GBS. For public breeding programs in the CG and national systems, low-cost uniplex SNP assays are about 15 cents a data point. So if you wanted to use a sort of a cascading genome profile approach where you develop your profile from a 200 SNP markers, that would cost you about $30 still for that profile, assuming you had the bioinformatics in place. Really, the costs per DNA sample for profiling need to be less than $5 for application in forward breeding. And that's going to take a real change in the way we do genotyping in our organizations. It's going to take industrial scale DNA extraction, very high throughput uniplex assay systems, imputation systems that exploit pedigree data. Yeah, we're really need to gear up in particular on DNA extraction, which is something of an artisanal process in most of our programs still. I've learned a lot this week from the companies and service providers that have come in to discuss how we can bring genotyping costs. One thing I learned is that we need to automate everything, but for us to benefit from very large upfront investments in automation, sample throughput is going to have to be very high. And that means we're going to have to aggregate sample numbers and standardize sample handling across locations, crops, and even centers. And it's really a question of whether the CGIAR and other public programs will be able to raise their game on management and coordination to allow this aggregation. If we don't do that, we're not going to reduce our costs to the level that we need to. There are a few open questions that have come up in the discussions. We had a very nice presentation from LGC on the pros and cons of in-house or outsourced genotyping. Either could work if we continue with in-house genotyping. The question is the CGIAR willing to coordinate across programs and centers? Can CG centers actually provide a high level of service at low cost? That's not, something that public institutions are normally strong at. Are public sector programs willing to build in depreciation into sample costs charged to programs? Because the technology turnover in this field is very, very rapid. But if we outsource, our service providers able to guarantee a turnaround time that's fast enough for these very aggressive breeding schemes that have been outlined to us today, where really, you may have 2 or 3 weeks to make a selection decision after harvest. Another interesting question that I've been learning about from a number of people here is whether sequence-based or SNP-based genotyping is what we need for sort of the frontline production genotyping. Ideally, a breeder would want a low-cost genome profile that gives information on every locus of interest from one DNA sample. But currently, that type of profile from GBS or Uniplex assays, before we have a high-throughput solution in place, are $20 to $30 and have complex bioinformatics requirements. So the question really is one question is how quickly will GBS costs drop and to what level? But another interesting point that was just raised with, Mike Olson ran through it with me just today, was the cost differential between an assay for, say, 4 diagnostic SNPs that are must-have SNPs, and the cost of a genome profile is a very important consideration. It's almost done. If you have, you want to genotype 1,000 lines per cross with pre-selection for, 1, 2, 3, or 4 diagnostic markers. It's quite interesting, I found, to pencil this out. Just one sec. The $20 profile with a $1 diagnostic assay for, say, 2 markers, that's the middle line there. The total genotyping cost for that set would be $6,000 if you did the diagnostic assay first, $20,000 if you didn't. Even if the profile is $5 and the diagnostic assay is $1, the savings is very substantial. You would pay for your machine fairly quickly with savings like that. So to conclude, we can double the rate of genetic gains by modernizing cultivar development. Discovery is necessary but not sufficient. To apply genomic knowledge in cultivar development, breeders really need high-quality information management, decision support tools. Genotyping profiling costs need to drop about 80%, and we need very good options for diagnostic assays. This is going to take a new way of organizing how we do business in the CGIAR. And the Gates Foundation is very strongly supporting the application of your science to cultivar development for the poor. We congratulate you on your progress and look forward to working with you. Thanks a lot.
Thank you, Gary, for a good presentation. Open for discussion, please. You can have some 2 or 3 questions. Yes, please.
My comment is we need to ask the national program, NARS breeders, are they able to use this? Will the cost they will meet? For example, many countries, they don't get any money for this sort of thing. So if there are any breeders in this group, particularly not from CGIAR centers, I think we should ask this question. I think that's the starting point.
If I can respond to that. It's up to the genomics scientists to come up with the solutions to reduce the costs and to provide breeders with very clear understanding of how they are going to benefit from the tools and the new breeding approaches. So it's up to us to explain. It's not up to the breeders to come to us. In terms of not receiving money for this, genotyping is a core breeding function. It is not an add-on. So in the same sense that running plots is a core breeding function, the value of genotyping is so great now that it simply must be part of the budget. You would have to reduce in other areas to make sure that at least for quality assurance, you are genotyping. Simply, estimates that I hear are that roughly 10% of the materials that you've got in the seed bag are not what you think they are in many breeding programs. Just eliminating that error will pay for the genotyping.
Any other question, please? Yes.
Gary, I think you're missing point 6. I was wondering whether it had anything at all to do with capacity. You talk about reducing breeding cycles. And I ask, when you look at the young breeders in this room, I believe in their early 30s, mid-30s, in 2050, they will be in their late 60s. What are we doing about capacity for the future? How are we equipping those we are training in universities, the tools they need to develop new varieties? I think we must rethink our model for increasing productivity in Africa and also in Asia. Training programs need to be properly organized, restructured, and I would wish that we would rethink this whole model.
You're very right, Eric. I've always been struck by the fact that the training of plant breeders has really very little to do with what they actually do in cultivar development. They do a genetic analysis experiment, and then they are sent out to become plant breeders. And cultivar development is different than genetic analysis. So it's very important that people like you reorient training so that there is a very strong focus on cultivar development for people who are going to go that route. They should be taught how to do what they say they are doing. And it's even more of a problem right now because the technology is changing so fast. The way plant breeding was conducted from the 1940s and 50s through to when I was in graduate school didn't change very much. It's changed a great deal since then.
Thank you, Gary. I believe there are no more questions. We thank you profusely for a presentation of a great significance. Thank you.